All right, this video is on eukaryotic gene regulation. Just kind of refreshing our memory on just gene regulation that's in prokaryotic cells. For example, if I have my DNA strand, which is, say, right here, okay, there are certain regions on that DNA, okay, specifically this little section right here, right, known as a promoter. This is where the RNA polymerase is going to attach to the DNA, and then we have these little segments of genes right here, right? Well, prokaryotic cells, right? So here's my gene, right? So the RNA polymerase is going to attach to the promoter, which then signals the transcription of this gene. Let's call it gene. Ooh, something original. A. Okay, so there's gene A. It's going to, the polymerase is going to attach to the promoter, which activates the transcription of gene A, and then that gene A will eventually go into the gene transcript, the RNA, the mRNA now, will eventually go to a ribosome, and then that RNA transcript, or that sequence, gets coded into a series of amino acids that we call a polypeptide. And that polypeptide will eventually become a protein, a functional protein, getting into the secondary, tertiary, and quaternary stages of proteins. Okay, so how eukaryotic cells are different, they're just different in detail. They're not necessarily different in practice. Okay, so we still have this DNA strand, right? And we have a promoter still, right? We still have this section of DNA that is going to allow the RNA polymerase to attach. Now, near the promoter, okay, you're going to have sites for different enzymes to come and attach, right? Well, the main, and the reason why these different enzymes, such as RNA polymerase, that's just one. In eukaryotic cells, there are numerous enzymes that are to come in here and actually start the translation process, right? Here's my gene right here. Gosh, what did I say? Gene C. Hey, I got you, didn't I? Now, so here's my little promoter, right? And this promoter in most eukaryotic cells is either near, or the core idea, or the core nucleotide sequence for a promoter in eukaryotic cells is known as the Tata box. Okay, those Tata box is obviously a series of adenine and thymine, just many A's and T's, A's and T's, A's and T's, and that is usually the principal promoter in many eukaryotic cells. Okay, so here's my RNA polymerase. The RNA polymerase may be the first to kind of attach, right? So here's my RNA polymerase, RNA pol. Right, there's the RNA polymerase. It may be the first to attach to the promoter, but you're also going to have, before this process even occurs, you're going to have a whole bunch of other little proteins that come along and attach and kind of reinforce the decision to transcribe this gene, which will eventually become a protein. All these little protein guys, okay, transcription factors, right? That's what the general name for all these different proteins, all these accessory proteins that come along and attach to RNA polymerase. Okay, so those are known as transcription factors. Now, this is kind of the pre-transcription regulation for eukaryotic genes. Okay, there are also post-transcriptional regulation. Okay, there we go. The main type of post-transcriptional regulation. Okay, let's go to post-transcriptional regulation. So this is happening after transcription has occurred. So my RNA, which I will now make green, right? There's my RNA. It's no longer DNA because its transcription has already happened. Okay, we can actually regulate this process in addition to regulating it before it happens. Okay, there's a couple key factors here that are involved in pretty much, I guess the best way to put it is stopping this mRNA from ever reaching a ribosome. The two regulators that we're going to focus on is um, miRNA, which stands for microRNA, okay, which is related to a process called RNAi, or RNA interference. Okay, and then the other is a protein, um, which is called a proteasome. Now, this is actual digestion of the protein itself, not just the RNA. So, meaning this is actual breakdown after not only transcription but also translation. Like say we made too much of a certain protein. A proteasome is a complex of proteins that'll come along and just chop up and digest the proteins that have been made. Okay, there is an important molecule that is involved in the action of a proteasome. It pretty much acts like a little marker to say, hey, this protein needs to be destroyed, and that is of course another protein. And that protein that attaches to the soon-to-be dead protein is called ubiquitin. Let's see if I spelled that right. Yeah, hey, good stuff. So, ubiquitin is like the flag. It's going to mark the protein bound for death. Okay? Now, that's, like I said, that's post-transcription, but it's also post-translational. I'm going to put that to bed right now because I really want to focus on the coolness that is RNA interference and the use of microRNAs. Okay? So, there is a sequence of RNA, okay, that are known as microRNAs. And they're just small segments of RNA that are going to actually bind with the mRNA that has been made during transcription. Let's go ahead and make this look a little more. Well, I don't know if you can make it look any better, but let's see here to show that this is actual. The mRNA is here. This one's bound for transcription or translation, and then this right here is my my micro RNA, which is blue. And what is going to happen right here is the concept of RNA interference means this piece of RNA right here is going to interfere with the transcription, or excuse me, the translation of that mRNA right here. Okay, and what it really does is by binding with, because I mean they're going to have A's and U's and C's and G's, right? They're going to they're going to bond, they're going to pair up with hydrogen bonds right here, right? Well, when that happens, that automatically tricks the immune system within, like the immune proteins within the cell, to go, that's double-stranded RNA. Double-stranded RNA doesn't belong in us because double-stranded RNA means virus, right? Oops. Right? So it pretty much, by that small, that microRNA coming in contact with the mRNA, it kind of fools some proteins inside the cytoplasm to think that it has been infected with a virus, and then here comes, big dog protein. It's going to come over here and it's going to chop up that mRNA. So when that mRNA gets chopped up, it never gets translated. Okay, so I mean, there's a couple different ways that we can translate or regulate our RNA sequences. Okay, one is before RNA is even made, right? We can actually have, we go back one, we have the transcription factors and the RNA polymerase that attach to the TATA box, which is, like I said, it's your main primer, your main promoter, excuse me, right? Right? That's your main promoter right there. And if we have no attachment of these transcription factors, or we have no, like a blocking of the TATA blocks or anything like that, that will result in a regulation of that gene. That gene never gets transcribed. But if we have this mess that I have right here, we have post-transcriptional regulation where we can have some mRNA get tricked and gets paired up with some microRNA, and that microRNA will trick the cell into thinking that it's been infected with a virus. So some digestive proteins will come along here and break apart the phosphodiester bonds and break apart the hydrogen bonds and pretty much completely extinguish that mRNA, right? And then we even have a post-translational regulation where a proteasome will pretty much pick up a protein that has been tagged with ubiquitin, right? And then that ubiquitin kind of waves a little flag saying, hey, this protein needs to be destroyed, and the proteasome will digest it. Okay, so there's a lot of different ways that we're able to regulate genes outside of just stopping transcription from ever starting. Okay?